Across a cold seafloor, a giant isopod walks toward the scent of a fallen carcass. Food may not arrive again soon, so this encounter governs more than a single feeding. The animal must locate scattered meals, consume enough, and endure the interval afterward. Its mystery is how a crustacean thrives where abundance arrives in brief, unpredictable pulses. If you watched from a lander, long stillness could suddenly become purposeful movement. Chemical cues spread through bottom water, turning a distant fall into navigable information. The isopod follows that plume with jointed legs and sensitive appendages. At the source, powerful mouthparts process tissue, while an expandable gut receives an exceptional load. Then metabolism and low activity stretch stored energy through scarcity. This investigation follows one rare meal from detection to digestion and renewed waiting. It treats fasting ability as adaptation, not evidence that food is unnecessary. Captive records show remarkable intervals without feeding, but captivity cannot define every wild schedule. Baited cameras reveal rapid aggregation, though bait creates an artificial concentration. Stomach contents and anatomy fill other gaps without reconstructing an entire lifetime. The carcass settles into sediment, and the surrounding plain begins to move. Giant isopods belong to the genus Bathynomus, a group of large marine isopod crustaceans. Multiple species exist, and reliable identification requires careful morphology or genetic evidence. They inhabit deep continental margins in several ocean regions, often on muddy or mixed bottoms. Their flattened, segmented bodies carry 7 pairs of walking legs and protective dorsal plates. Compound eyes and long antennae gather information where sunlight is absent. Low temperatures reduce metabolic demand, while deep-water darkness removes the need for daytime shelter cycles. Large size may improve energy storage and movement across distances between patchy food sources. This interpretation is plausible and widely discussed, although gigantism probably has multiple influences. Bait enters the water, and dissolved molecules spread downstream with local currents. An isopod detects the chemical gradient, turns into the plume, and advances toward increasing concentration. Directional movement converts smell into access before competitors consume the source. Arrival timing, therefore, has a direct consequence for energy gained. When you see many isopods around bait, the gathering demonstrates attraction, not ordinary population density. The camera station has concentrated odor and food into one small artificial location. Field interpretation must separate response radius from the number normally occupying each square meter. At natural food falls, giant isopods can scavenge fishes, cephalopods, and other animal remains. They may also consume living prey when opportunities permit, so scavenging is not necessarily exclusive. Observed feeding shows mouthparts tearing or rasping accessible tissue from a larger resource. The foregut and digestive system accommodate substantial intake during a profitable encounter. A large meal expands the body between rigid plates, visibly changing its outline. That expansion stores material for digestion after the feeding crowd disperses. The sequence joins rare opportunity, heavy consumption, delayed processing, and prolonged survival. It resembles feast and famine, but each phase remains controlled by physiology. Scarcity becomes sharper after the odor plume fades. The isopod can reduce activity, limiting muscular expenditure, while digestion releases absorbed energy. Cold ambient water further lowers biochemical rates compared with warmer, shallow habitats. Captive Bathynomus have survived long fasts, demonstrating physiological tolerance beyond ordinary meal intervals. Those records establish capacity under managed conditions, not typical fasting duration on the seafloor. Wild individuals may encounter small food items that cameras never record. They may also move beyond surveyed areas, making absence from bait stations ambiguous. A stomach that appears empty records one moment, not the full interval since meaningful nutrition. Stable biochemical tracers can integrate diet over longer periods, yet they rarely identify each meal. Evidence, therefore, supports intermittent feeding while leaving natural frequency incompletely quantified. The word thrive must also be used carefully. Survival after fasting is documented, but population growth requires reproduction, juvenile recruitment, and suitable habitat. An adult enduring scarcity does not prove every life stage experiences equal resilience. Egg-bearing females invest energy in large broods carried within a ventral marsupium. Developing young rely on maternal investment before emerging as mancae, resembling small adults. Reproductive timing and frequency remain poorly known for many Bathynomus species. A rare meal could support future reproduction, but direct energy allocation has not been fully tracked. Likewise, extreme fasting in aquaria may reflect stress, food preference, or altered environmental cues. Researchers should not treat refusal of offered food as a perfectly natural feeding calendar. The strongest conclusion comes from convergence across anatomy, feeding observations, and metabolic context. Giant isopods are equipped to exploit large pulses and tolerate long gaps. Exact wild intervals vary with location, season, currents, and the supply of falling organic matter. Competition adds another constraint when a carcass reaches bottom. Fishes, amphipods, hagfishes, crabs, and microbes may consume the same temporary resource. Early arrival lets an isopod feed before soft tissues disappear. Crowding prompts repositioning, gripping, or pushing as individuals seek exposed edges. Protective plates reduce injury, while flexible joints preserve access to the carcass. These actions convert armored form into success within a short feeding window. Afterward, fragments and waste feed smaller scavengers and sediment microbes. The rare meal thus spreads energy through a community rather than ending inside one large consumer. If you follow the carcass downward, its carbon connects surface production with deep benthic life. Giant isopods help accelerate that transfer by opening tissue and redistributing fragments. Their movement may also disturb surface sediment locally while they search and feed. Such ecological effects depend on abundance, carcass size, and the surrounding scavenger community. Bait experiments can compare arrival sequences, but artificial bait type influences which animals respond. Currents control odor dispersal, so response time cannot be translated directly into distance. A fast arrival might mean nearby animals, strong flow, sensitive detection, or several factors together. This limitation matters when estimating populations from camera footage. Human activity adds new pulses that differ from natural food falls. Fishing discards can provide carrion, while lost gear can trap scavengers attracted to bait. The scent cue triggers approach, but enclosed traps can convert feeding behavior into mortality. Bottom trawling may capture isopods and alter the sediment habitat they cross. Deep warming and oxygen change could raise metabolic costs or compress suitable zones. An animal adapted to slow energy expenditure may be vulnerable when maintenance becomes more expensive. Rare meals remain useful only when stored energy can outlast the next interval. The isopod's success, therefore, depends on both feast size and environmental stability. Environmental stability ultimately determines whether stored energy can bridge each unpredictable interval. Long-duration landers could record unbaited movement, then compare responses after controlled odor release. Telemetry may eventually reveal travel distances, activity cycles, and return to feeding sites. Respirometry, under realistic temperature and pressure, could quantify metabolic change during digestion and fasting. Biochemical tracers could distinguish recent carrion from longer-term dietary sources. The decisive pattern would link heavier feeding directly with longer survival and later reproductive investment. These four approaches would connect behavior, energy use, movement, and food history. Species identification must accompany each record because Bathynomus diversity can complicate comparisons. The needed seasonal pattern must show whether meal pulses track fisheries, migration, or surface productivity. If you return to the opening carcass, the first arriving isopod is no mindless glutton. Its antennae have converted a fading chemical trail into a precise approach. Its mouthparts and expandable body turn temporary abundance into stored opportunity. Its low activity and cold-adapted metabolism extend that opportunity through the quiet afterward. Some wild snacks will remain unseen, and exact fasting intervals remain unknown. Still, field behavior and captive physiology support the same central strategy. Bathynomus thrives by finding dispersed food quickly, feeding heavily when possible, and spending energy with exceptional restraint.